Hello everyone, welcome back to my video tutorial for single-cell RNA sequencing data analysis. Today I'm going to show you how to integrate spatial sequencing datasets. Currently for spatial single-cell RNA sequencing data preparation, the tissue size is limited by current technology. I'm going to show you how to integrate a spatial dataset for the mouse brain tissue. You can see here, in order to connect all the spots from the brain sections for single-cell RNA sequencing data preparation, we have to separate the tissue into two parts, frontier and posterior, and do separate spots collecting for the sequencing data preparation. But thankfully, packages were developed to integrate the spatial dataset. Today I'm going to show you how to use Scanorama package and integrate the spatial sequencing data for the mouse brain sections. Actually, I looked at the original publication. I couldn't find the abbreviation for Scanorama. So we just call it Scanorama and use it for today's analysis. I'm going to use the Jupyter Notebook again. First, you need to install the package Scanorama. You can just use pip install. I installed the package already, so we can go to the next step and load the packages. We also set the parameters for today's analysis. So next we can read the data in. First we can read the anterior dataset. For the first time, if you run this code, Python will download the data in your computer. I ran the code once already, so Python will read the data from my computer. So let's read the anterior data in. Then we can perform quality control analysis. First, we can calculate the percentage of mitochondrial DNA. Then we can use the violin plot to see the N genes by count, total count, and percentage of mitochondrial DNA. So you can see the violin plot for the N genes by count, total count, and percentage counts for mitochondrial DNA. This is the same dataset that I showed you in my previous video, how to analyze spatial single-cell RNA sequencing data. So we can just use the same parameter to subset the anterior dataset. So next we can read the posterior data in, and also perform the quality control analysis. We calculated the percentage mitochondrial DNA, then plot N genes by count, total counts, and percentage mitochondrial DNA for the posterior dataset. The data looks very similar as the anterior dataset, so we can just use the same parameter to subset the dataset. So we read both dataset, the anterior and the posterior data, and also performed the quality control. Now we can normalize both dataset and find the variable genes for both dataset. We can run both functions together for both datasets. So next we can prepare a data list contain both the anterior and the posterior dataset. as the data list, we name it as the A datasets. So let's have a look at the A datasets. You can see we have two datasets in the data list. The first one is for the anterior dataset. We have 2,500 cells and more than 17,000 genes. The second one is for the posterior dataset. We have 2,908 cells and nearly 17,000 genes. Now we can use the Scanorama function to correct the batch effect from both datasets, and also we only keep the genes that are presented in both datasets for integration. Let's run the first Scanorama function. Okay, it is done. You can see Scanorama found 16,486 genes between both datasets. And we named the data as A data as collected. So let's have a look at the data inside the A data as collected. You can see the collected dataset still contains two datasets, the anterior dataset and the posterior dataset. Probably you already noticed that each dataset still has the same number of cells, but now both datasets have the same amount of genes. And also in the multi-dimensional observations, we have the Scanorama information stored in both datasets. Now we can perform the second step for the data integration. Scanorama uses the concatenate function to integrate both datasets. So the data will be the A data as collected, and also we use the library ID to label each dataset. Then we know the cells from the anterior dataset and also the cells from the posterior dataset. We are going to merge all the cells, but we cannot merge the image information from both datasets. So here we use unstructured merge as unique to keep individual image information in the integrated dataset. So let's integrate the data. Before we run next step, we can have a look at the A data spatial. Because after integration, we named it as A data spatial. Let's have a look at the A data spatial. Now you can see we integrated both datasets as one dataset, and the cell numbers have been added together from both datasets. And also in the multi-dimensional observations, we have the Scanorama information. Now we can compute the neighbors, UMAP, and find the cell clusters using the Naden method. Once again, I'm using the resolution 0.2 to reduce the total number of cell clusters. Okay, so we are ready to visualize the cell clusters. Use the plot function, and we are going to see the cell clusters in one figure and show the cells from both datasets in the second figure, because we use the library ID to label the cells. So from the UMAP plot, you can see on the left hand side we have 9 cell clusters from both datasets, and on the right hand side figure, cells from anterior dataset were shown in blue, and the cells from the posterior dataset were shown in orange. You can see, even we are integrated both datasets, the blue cells and the orange cells were not completely mixed together. This is because we have distinct cell types in different regions of the mouse brain. For example, the posterior data has cells from the cerebellum. Now we have the cell canisters. We can project the cell canisters onto the brain sections, use the information stored in the spatial coordinations. Because we cannot merge the images from anterior and posterior, but the cell canisters could be presented on both images. So we want to keep the same color for each cell canister on both images. So let's save the canister color from here and use the same color on both images. We can have a look at the information in the canister colors. You can see we have 9 canisters here, and we save the color code for each cell canister. Then next we can use the color information for each cell canister to visualize them on both images. So we can visualize the cell canisters on both sections together. We project the cell canisters for the anterior sections on the left and project the cell canisters on posterior sections, and we put the posterior sections on the right hand side. So let's plot both images together. You can see we have 2 images. The anterior image is on the left hand side and the posterior image is on the right hand side. And also we projected the 9 cell canisters on both sections. So we integrated the anterior dataset with the posterior dataset, and now we can visualize them on the whole mouse brain tissue section. And you can see, use the package Scanorama, we integrated both datasets very well. You can see the blue cell canisters in both images. They are in the cerebral cortex area, and also in the ventral region you can see the purple cell canister. It is presented in the ventral region, in the anterior section, and also in the posterior section. After integration, we can consider them as one dataset and also show all the cell canisters on the whole mouse brain section. Now we can do more analysis on the whole brain tissue section. For example, we can identify the mark genes for each cell canister on the whole brain tissue. I introduced the rank genes group function to identify mark genes for each cell canister. I can use this function as an example to show you how to do further analysis. We will use the Wilcox method to identify the mark genes for each cell canister. Okay, it is done. So we can use dot plot as an example to show the mark genes, and we will plot 8 genes for each cell canister. Okay, we have the dot plot. So you can see the cell canisters were labeled on the left hand side, and the mark genes for each cell cluster were labeled on the bottom. And we can use the spatial plot function to show some mark genes for different cell clusters. For example, we are going to show the PPP1R1B mark gene for cluster 3. It is a mark gene to label dopaminergic neurons in the midbrain, so we can just change the color to the gene name and show the spatial plot for this gene. You can see PPP1R1B is highly expressed in this region. It is the cluster 3 in the midbrain. We can also use another mark gene called PCP2. It's a Purkinje cell mark gene. We know Purkinje cells are in the cerebellum. If we make a plot for this gene, you can see PCP2 is highly expressed in the cerebellum, and it enables the Purkinje cells in the cerebellum. So I just use the rank genes groups information function as an example to show you how to analyze the integrated dataset. You can do more analysis, use the integrated dataset. So that's it for today's demonstration. I hope my video can help your spatial data set integration. If you like my video, please subscribe my channel and share my videos with your friends. Thank you and see you in my next video tutorial.